name's Alison van Eenennaam and I am a professor at UC Davis, and I am out here at the UC Davis beef barn with my friend Prince and Cosmo. And Cosmo is a pretty special guy, the black one over there, because he is the first genome edited knock-in bull calf that's ever been produced using gene editing in a bovine embryo. So he's a pretty special guy. And he's pretty special because he's predicted to produce 75% male offspring. So normally you get 50-50. If you get a Y from your dad, you're male. If an X from your dad, you're female. But he's got an extra copy of the gene that makes you go to become a male on one of his non-sex chromosomes. And so we think that the animals that inherit that, in addition to the XX, so what would normally be a female, will actually be triggered down the male pathway and will grow up as males. And males in beef production are actually more efficient at converting feed to gain. There are no other cows like Cosmo in the world. He's kind of unique. A healthy 110-pound bull calf, named by Joey for the green glowing character from Fairly Odd Parents. The only thing left to do was make sure he actually had SRY inserted into chromosome 17 and to check if he was XX or XY. PCR analysis was performed on DNA derived from his blood, and all of the results showed the same thing. He was a heterozygous SRY knock-in XY bull calf. The team plans to produce offspring from Cosmo to eventually answer the question of whether the inheritance of SRY in XX cattle will result in males. It's about a 5-year research project, so we had to optimize a way to get the gene to knock in the developing embryo. And normally embryos don't have a lot of repair pathways turned on, and so we had to kind of trick the embryo into replicating the gene that makes you become a male, the SRY gene, on the autosome. The trick was we actually added a gene called green fluorescent protein adjacent to the SRY gene so that we could fluorescently screen the embryos before we did embryo transfer into cows. And so we could basically only transfer the gene or the embryos that had the SRY knocked in. My advice to students would be, pursue research that you're really interested in, because there's going to be a lot of days when things don't work in the lab, and if you're not doing research that's interesting to you, you're not going to be very happy. But I think that you never know what you're going to end up doing. When I was a young person at college, they hadn't even invented editing or genetic engineering. They'd only just discovered the DNA structure, and so I had no idea this would be my career because this job didn't exist yet. So be open to new discoveries, and there's so much exciting things going on in molecular genetics at the moment that I think it's just such a fascinating field and very fast moving. So hang on to your horses if you want to get into it, because it's going to move fast. I think UC Davis is the best agricultural research university in the United States, and I might even argue the world. And there's so much opportunity here to do cutting-edge research that's both really exciting scientifically, but also has application to the field. And my real interest is translational genomics, or taking science to the farmers. And that really, I think, is the opportunity that is available at UC Davis.
Yeah, the future of agriculture, I think that unfortunately agricultural research kind of has this reputation of being, someone in overalls chewing on a hayseed or whatever. And it's super sophisticated, and there's so much opportunity to do precision agriculture and just seriously, everyone's always going to need to eat. And one of the most pressing issues we have is trying to continue to produce food for a burgeoning population whilst minimising the environmental footprint. So there's a huge societal good from that, and also really challenging science. What species was the first successful CRISPR livestock experiment done in a food animal species.